perform statistical analysis using the tameNMR toolset, you need to first prepare a factor template. This allows us to see the order in which tameNMR parses the data. This can then be exported via the Save icon and opened in a spreadsheet. Here we can see there is one column that gives us the full title of each spectrum as tameNMR perceives it. What I've done here is add another column in where you can actually see the order of the numbers. tameNMR thinks like a computer, and therefore these are not in the order that we would expect. We can also add in the information from the study. Going to the samples description in the metabolites library held at the EMBL EBI, we can see the sample name and also the grouping. In this case, we're looking at the oxygen level, and we can therefore input this information into the second column of our factor template. Save this as a tab-separated volume and upload it to tameNMR. Here is our uploaded factor template, which we are now going to use to perform a t-test. Going to the Statistics module, select Students t-test and select the data you wish to perform the t-test in. Now this should be the data that's been binned for the data set that matches the factor file. This is the cell spectra that has been binned, and we're using the cell factor grouping table. You will then be prompted for which factor column to use. If you look again at the factor data set, what you end up with is 3 columns. However, the first column is disregarded, as stated here. Therefore, the first column after the names is what we will be using. These data sets are not paired, so we can now proceed with the student's t-test. Once completed, we can see how many of the buckets appear over the threshold we've set for p-values. We can also see how many both before and after p-value adjustment. In this manner, we can do a quick t-test on our results to understand how the data has come about. tameNMR is funded by the BBSRC grant to the University of Liverpool in collaboration with the NMR Metabolomics Facility and the Computational Biology Facility. You can reach us via Twitter, or you can access other tutorials on tameNMR here.